Good morning, everyone, and welcome to today's MPG Primer series. We are very excited to have another tutorial today by Ramin Shirali. He's a graduate student at the Delft Bioinformatics Lab within the Faculty of Electrical Engineering, Mathematics, and Computer Science at TU Delft in the Netherlands. He is just on the final stages of completing his PhD and will next start a postdoc at the Leiden University Medical Center with Eric van de Akker in a couple of months, where he will work on acute myeloid leukemia. We're very excited to have him here today to talk about haplotype assembly using short and long reads. And we'll be posting some links that will be able to have you have primary access to the code if you want to follow along as you watch this either live or during as a YouTube session. So thank you so much, Ramin, for joining us all the way from the Netherlands today, and we look forward to your talk very much.
Hello, everyone. I'm Ramin. I'm a last month PhD student at TU Delft. And again, thank you for having me. Today, I'm going to talk about how to assemble the haplotypes using short and long reads. Just to begin, so most of you probably know that we humans have two copies of each chromosome. One of them is coming from our mother, the other is coming from the father, and we are called diploid because we have two copies of each chromosome. But some organisms have more. For example, potatoes are tetraploid, and they have 4 copies of each chromosome. And some organisms, like some yeasts, have different copies for different chromosomes, and they're called aneuploids. So the problem of haplotype assembly is like in contrast to normal de novo assembly, where we have the haplotypes, we sequence them, and we construct a single sequence based on the reads, which is a mosaic structure of the haplotypes. So it has some of the alleles of one of the chromosomes and some of the alleles of the other chromosome. In haplotype assembly, we try to separate these two haplotypes and basically reconstruct the sequence of both of them from a shared sequence, from the shared read pool. And there are many advantages in doing that. For example, in this study, they are showing that if you look at the single SNP, it's not enough to find the markers that are related to yield in wheat. And if you only reconstruct the haplotypes of very conserved regions, you can actually use that to increase the yield of wheat and use that in breeding programs. So today I'm mainly going to talk about HAT and nPhase. Both of them are very recent tools for haplotype assembly. They both use short and long reads. They are both based on SNPs. HAT is developed by us at TU Delft, but nPhase is also a very, very cool tool to use. So I will also go through that. They are taking different approaches. So nPhase is trying to cluster the reads based on the similarities that they have. So it's an iterative algorithm. It starts with reads and it tries to merge the reads so at every step, they merge the two sequences that are the most similar to each other. And they try to reconstruct the haplotypes based on that. HAT, on the other hand, take a seed and extend approach. The main difference is that nPhase use short reads only for variant calling, and it doesn't use them for reconstructing the haplotypes. But HAT use short reads for variant calling and also use them for haplotype reconstruction. So just a little bit more detail. As I stated earlier, nPhase try to merge the most similar sequences at every step. So, for example, if you look at the two reads at the bottom, they both have white allele at this SNP. So these black ones are the SNPs, and if these two reads both have the white allele at these locations. And these two at this step are probably the most similar reads together, and it kind of merge them together and create a longer sequence. And for determining the allele that this larger sequence should have at these SNP locations, it just takes a majority voting between the sequences that are merging. As I said earlier, HAT, on the other hand, take a seed and extend approach. That means that it starts with a very small regions. It tries to understand the alleles that each haplotype has at those small locations, and then try to use those small regions as a base to reconstruct the whole haplotype. So it tries to merge them together and like extend them in a way that it covers the complete haplotype. So and the way to do that, the way to create seeds is that it look at the reads. So because the reads are coming from a molecule and they are originated from a haplotype. So like if you check that the alleles that the read has, we know that, oh, these alleles are definitely together in the same haplotype. So we can immediately link them together and phase a very small region. For example, here at the left, we know that the A and C are from the same haplotype, and C and G are from the same haplotype because they are together in a single read. And that can be the starting point for reconstructing the haplotypes. So the main difference that HAT has compared to other seed and extend approaches is that it first tries to detect the multiplicity blocks. So if you look through the chromosome, there are some regions that the haplotypes are actually identical and there is no difference. There is no significant difference between them. And it's a waste if you try to actually reconstruct. Actually, it's impossible to reconstruct the haplotypes of those regions, especially because they are identical. What does it mean they have the same sequence? And if these regions are large, it's impossible to connect the phase regions on the side of them. So, for example, at the start of this chromosome, these three haplotypes are different, but then they're identical at some location. And if this region is huge, it's impossible to link what you have here to what you have over there. So what HAT does is that first, based on the seeds that it creates, try to find these ploidy blocks, try to find at each region it can reconstruct three haplotypes, at each region it can reconstruct two haplotypes. It actually ignored the regions that there is only one haplotype because that's basically the reference sequence. And then it tries to reconstruct that number of haplotypes for that region. So like, that's it. But that's what what is different from all other haplotype assembly tools. And we found that very, very useful because we were often failing to reconstruct the chromosome scale haplotypes. Well, it's understandable why we were failing to do that, because it's actually impossible. So why we are trying to do that? It's better to take a step back and only phase the regions that are required to be phased. And well, this is just a multiple, just to give you a better picture. This is the multiplicity blocks of a real chromosome. So this is chromosome 2 of CBS 1483, which is a yeast strain. At the bottom, you see the SNPs between the haplotypes. And the black boxes here in the middle are actually the seeds. And the red box on top of them is showing that there is this multiplicity block with multiplicity level of 2. That means that we have 2 haplotypes only here. So although this chromosome is a triploid, it's a waste to try to reconstruct 3 haplotypes for these regions. And the regions that you don't see anything that means that these regions are mostly identical. And if you look at the SNPs, you see that we have some SNPs, but they're so far from each other that it's impossible to connect them together, even with the long reads. So there is no point in trying to reconstruct the haplotypes in these locations. And here at the right and on the far left, we have also like few multiplicity blocks with multiplicity level of 3. That means that we can reconstruct 3 haplotypes for these regions. I'm going to talk a little bit about the results that we got, and then we go to the tutorial. So we simulated 6 data sets from symbiosis 1483 chromosome 2. We simulated short and long reads and also haplotypes. So we used haplogenerator to simulate haplotypes, ART for simulating short reads, and BAD-READ to simulate long reads. And CBS 1483, I'm talking a lot about it, but you are, well, you probably don't know the name, but you are familiar with it. It's a close relative of the strain that is used in Heineken beers. And it's aneuploid, so that's the main reason that we chose this strain for our study, because it had a different level of ploidies, and we could use them to model like different level of ploidies and to simulate our haplotypes. So as I said earlier, we simulated 6 data sets. With different level of ploidy, with triploid, tetraploid, and pentaploid, and different level of heterozygosity. So the ones that have low heterozygosity, that means that they have the same amount of SNPs as the actual haplotypes in the real data set that we had. And the ones that have high heterozygosity are created with the model that we created based on the real data set. So like we try to fit a log normal distribution on the SNPs, distances between SNPs of the real data set, and we use that model to simulate the SNPs in the high heterozygosity data sets. The reasons that they have more SNPs than the low heterozygosity data sets is that the simulator that we were using was not actually simulating the variation deserts. This is what I'm calling a variation desert. If you look at here, there are some regions, yeah. So the model was basically modeling these parts that the SNPs are very close to each other. But at some point there is a huge variation deserts where you don't see any SNP. And the simulator was not modeling that. So that's why they have more heterozygosity. And then we reconstructed the haplotypes and with both HAT and nPhase, and these are some of the results. So both HAT and nPhase can cluster the reads based on the final phasing that they have. If you look at the results for most of the data sets, except for the triploid high heterozygosity data set and the tetraploid high heterozygosity data set, HAT is clustering more reads, and it also has less error. So it's kind of doing better, but I want to again point out that nPhase is also doing great. So if you compare, I don't have the numbers here, but if you compare them to some older methods, they are really outperforming them. But HAT and nPhase are really close to each other. And if you also look at the phasing performance, like because these are simulated data sets, and you can actually compare the alleles that we assign to haplotypes to the alleles that the actual haplotypes had. If you and then you can calculate the accuracy based on that. If you check the accuracy, HAT is outperforming nPhase in all of them, but again, nPhase numbers are also very brilliant. But then we also used the HAT to reconstruct the actual haplotypes of CBS 1483 on the real data set, and the results are I don't know if I should call it less impressive. The thing about it is that like there are some of the chromosomes that are, although there are like for example, look at SE1. It's a diploid, but it has only 21 SNPs. So like the chromosomes are highly identical. And yes, we are phasing less than 1% of the whole chromosome. But that's it. The reason is like there is nothing to phase. So it's also that's the thing. But if you look at the chromosome SC1, which has more heterozygosity, we can actually phase up to 54% of the chromosome. And we also wanted to some more validation for the phasing. So like we took a better look at what had created. So we took a better look at this small ploidy block that we have for chromosome 2. And we basically mapped the short reads and the long reads of haplotype 1 to the haplotype sequence that had created for haplotype 1. And we also aligned the short reads and long reads of haplotype 2 to the same sequence that had created for haplotype 1. And as you can see, the reads that belong to haplotype 2 are all sharing some differences with the sequence that had reconstructed with haplotype 1. That means that, oh yeah, all of these reads are sharing some differences. So it's kind of showing that they are coming from a different source, a different haplotype, that means. So let's get into the tutorial. So I have the commands here in the slides. So a couple of months ago, we organized actually a workshop for phasing haplotypes. And I shared the links earlier. There we have every command that you need to use to get the same results. In this study, like in this tutorial, we're also working on one of the chromosome chromosome SC1 of CBS 1483, which is a triploid. And for phasing, we use nPhase and HAT. So these are the links. I guess they're on the chat. They're in the chat, and like you can use them later on. And the data is here. You can download it. So the first thing, of course, I recommend to you create a Conda environment. So the Conda environment that we have here has all the packages that you need for this tutorial. It has both nPhase and HAT and like the assemblers, the alignment tools that you're going to need. So when I want to reconstruct haplotypes, the first thing that I look at is the reads. So first, I always use nanoplot to visually look at my long reads. So after you use a nanoplot with this command, and like you need to specify the output directory, but in the output directory you get a report.html file, which has a nice visualization over your reads. So in this case, about the data that I shared, it's like 60x coverage. And if you remember earlier, I said that it's a triploid. So that means that similar to our study earlier, it's like probably 20x per haplotypes. And the average read length is like 10 KB. And if you look at the read length distribution, you also see that the average read length is around 10K. But there are some, not many, but few reads that are less than 1,000 nucleotides. And in my experience, it's usually useful to remove those kind of those reads because they are first of all, long reads are error prone, and these reads, because they are shorter, they are not covering lots of SNPs, and it makes them more sensitive to the errors. Because like both nPhase and HAT always look for high coverage for the actions that they are taking, because when you are using long reads, it's always good to look for high coverage and high support because of the errors that they have. And then these reads are shorter and they are not covering many SNPs. So it's really hard to assign them to haplotypes. And you might assign them to wrong haplotypes because of the errors. So I use field long to remove the reads that are less than 1,000. Sorry, there is an error here. I wrote 10,000 here, but I meant 1,000. The next step is to align reads to the reference sequence. So I align the short reads with BWA mem. So first you need to index the reference sequence, then you can use BWA mem to align the short reads to the reference. As you can see here, my short reads are paired end, so I'm giving both pairs to BWA mem, and the T is just the number of threads. And then I convert the SAM file to a BAM file because it's much faster to use BAM file in the code. And then I also sort them because, again, a sorted BAM file can be indexed and it's much faster to access to it in the code. And then we use Freebase for variant calling. I personally, so you are free to use any variant color that you like, but I personally recommend Freebase. Also, if you look at the nPhase paper, they also recommend Freebase for variant calling. It's a very successful tool. And the -C2 here means that for every variant that you're calling, look at least for 2 short read support. So at least 2 short reads should have that variation. And the ploidy is, of course, obvious. It's the ploidy of the chromosome. And then both nPhase and HAT are SNP based. They only look at SNPs for phasing, for reconstructing the haplotypes. Actually, most of the polyploid haplotype reconstruction tools only look at SNPs for haplotype reconstruction. And then here we are using VCF filter to extract SNPs from the VCF file that Freebase created. And then we just zip that and then index it to have a faster access in the code. We also align the long reads to the reference genome with Minimap2. The minus secondary equal no means that only report the best alignment of the long reads to the reference genome. So only look for the best mapping and report that one. I find that usually useful for haplotype reconstruction. And again, we convert that to a sorted BAM file with the same method. And then we have HAT. Then you have everything to run HAT. So for HAT, you need the alignment of the short reads and the alignment of the long reads and ploidy and the VCF file. That's it. So for basic the basic usage of HAT, that's all you need. Well, the chromosome name is also a name, but and the output, but those are not like input kind of thing. So for the basic usage, that's all you need. But here I'm using the -HA command that actually tries to reconstruct the sequence of haplotypes at the same time. Well, it's not at the same time, but after the phasing, it uses the results and it uses mini-ASM to reconstruct the haplotypes. I personally don't recommend that. So for reconstructing haplotypes, I always prefer to look at the phasing output and look at the reads that the tool cluster, and then use my own tools. I personally prefer Fly for assembly. So I prefer to use my own tools for haplotype reconstruction. So I personally never use -HA, but some people want to have everything in one command, so it's also there. But if you want to use -HA to reconstruct haplotypes, as it is in this example, you also need to provide the long reads FASTA file and the short read FASTA file, like FASTQ file. So like the hat can go and extract the clustered reads from those files and give them to mini-ASM to assemble the haplotypes. And these two commands, -th and -ma, you probably never need to use that. These are for evaluation mainly. And so you can also give hats like the true haplotypes if you have them, if it's a simulated study. And then so based on the true haplotypes and based on the multiple genome alignments, hat report some scores. And then after using hat, these are the output that these are the outputs that you get. So in this example, it gives us 3 directories. So the first 2 numbers in the directory are the start and the end position of the ploidy block. So if we had another ploidy block in this example, there would be also another directory with the start and end position of that ploidy block. And the ploidy blocks do not overlap with each other. And but in this example, we only have like 3 ploidy block, 1 ploidy block. That's why they are sharing a start and end. But within the ploidy blocks, there were some regions that could not merge together. Actually, that's not that important, because if you look closely, and sorry, let me rephrase that. The third and the fourth number are start and the end location of the phase block that had created, like the region that it actually phased. So this is the whole ploidy block, but this is the region that it phased. And if you look at closely, you see that this is almost covering the whole chromosome. So our chromosome was like 211 kb. So the third block is actually covering the whole chromosome, and these two are very small regions right at the end, and they don't really matter that much. The reason that they don't actually matter is also that these coordinates are the starting and ending, so the location of the first and the last SNP. And if you look at the reads that had actually clustered, they go over these regions, so that the read can actually goes a little bit more to the right side or a little bit more on the left side. So that means that the reads that are clustered can actually cover these small regions that are a little bit more at the end of the chromosome. So they don't matter at all. There are 3 more files that are important. The first one, this PDF file, is the ploidy blocks. It's a figure. It's good to look at it. It's in the next slide. We will take a look at it. And this file, the phase read blocks, is actually like a file separated by ploidy blocks, and within each ploidy block, it's mentioning that which read belongs to which haplotype. So it basically assigning reads to the haplotypes, the reads that it can assign. So some reads are might be in the regions that there are not many SNPs there, and those are not assigned to any haplotypes. But the ones that are assigned, you can find them in this file. And the other file is the phase matrix file. This one is just mentioning what alleles had assigned to the haplotypes at each SNP loci. So the rows are haplotypes and the columns are SNP loci, and then there you can see the allele that is assigned to each haplotype. And then if you look at, because we use the -HA command and had to assemble the haplotypes, if you look at this directory, you can also see the assemblies. Well, as I said earlier, had to use mini-ASM to assemble the haplotypes, so the output is the GFA file, but you can convert that to a FASTA pretty easily. And as I said earlier, this is the ploidy blocks. Well, the ploidy blocks that we got for this tutorial. And as you can see, we have only one ploidy block. And that's what we could also see earlier, that like there is only one ploidy block here. And it goes over the whole chromosome and the reason that HAT could do that, and the reason that HAT could phase this chromosome start to end, telomere to telomere, is that there are lots of SNPs, like it's highly heterozygote. And as you can see, there are lots of SNPs between the haplotypes, and HAT could use them to reconstruct the haplotypes. And well, this is a command that you can use to reconstruct the haplotypes using nPhase. So nPhase can be run in 3 modes. So you can give them your own VCF file and alignment files and just use it to reconstruct the haplotypes. Or you can also just give it the reads and ask nPhase to do the variant calling and the alignment by itself. Well, in the core, it's also using BWA and Minimap2 for the alignment, and it used 3Base for variant calling. So we could also just feed them the data that we had from when we wanted to run HAT. But here in this example, I'm asking nPhase to do everything by itself. So I'm giving it the reference because it needs to align the reads to the reference for variant calling, and specifying the long reads and telling that it's Oxford nanopore reads, and I'm also specifying the short reads. So the output that nPhase gives you is 5 directories. Most of them are like intermediate files. You don't care about them. So the actual output is in the phase directory. And inside that, you have 2 other directories, plots and FASTQ. In the FASTQ directories, you can find the reads that are assigned to haplotypes. And because it's the nPhase gives the FASTQ file, you can immediately use that for assembly. And in plots, you can find some plots that kind of help you to interpret the results. I will go through them. And there are 2 other important files, the variance.tsv and phasedatasimple.tsv. The variance.tsv is telling you that for like the nucleotide, that the allele that nPhase assigned to each haplotype at each SNP loci, and the phase data simple also tells you the start and the end position of each cluster. So if you remember from back then I started the presentation, nPhase is kind of a clustering tool. It tries to merge the reads together and create huge cluster of the reads. And in this file, you can find the start and the end position of these clusters. If you look at the figures that nPhase creates, this is an important one. It actually shows where the location that each haplotype is, like each cluster is covering. So like this cluster is starting from here, it goes all the way to here. And on the y-axis, you can find the the coverage that it has for each base at the chromosome. I personally, so that's actually the main motivation that we develop HAT. So I find interpreting the results of HAT a bit hard. Interpreting the results of nPhase a little bit hard, because for some reasons it has more clusters than the number of haplotypes. For example, here or here. And it's not clear what should we do with the extra cluster that we have. So I can live with ignoring this cluster because it has a very low coverage. But this one has almost, the purple one, the dark purple one, has almost the same coverage as the blue cluster. So it's hard to ignore that. There is also another figure that tries to show which location each haplotype is, each cluster is covering again. But so it also includes the smaller clusters that were not included in previous figure, and that you can see the same problem that I mentioned earlier. There are regions that have more, that there are more clusters than the actual haplotypes. So what should we do with these two extra ones and with these little ones? Should we just ignore them? And this is actually a good example. I can also show you the results that we got for running nPhase on a little bit longer chromosome, 800 kb. And this is the figure. I'm personally scared when I see this figure. I don't understand anything from it. And yeah, which ones are the actual haplotypes? And if you look at the other one that tries to line them up, again, same problem. 4 haplotypes here, 4 haplotypes here, even more here. What should we do with the extra sequences? I don't know, and that's the problem. That's the problem that I have with nPhase. So the thing about HAT is it tries to reconstruct the same number of haplotypes as the multiplicity of that block, and that's very useful for me. So you can all like follow, so all of the commands. So I already shared their links, and I guess they're on the chat. You can follow all of these commands yourself. They are well documented in the link that I shared, and the data is also available. In the end, I would like to thank my colleagues at Delft, especially Aysun and Thomas, that helped a lot with this project. And if you wanted to use HAT or nPhase, or I can also help with WhatsApp. If you wanted to use any of these 3 tools for reconstructing your haplotypes, feel free to contact me. I'm more than happy to help. And thanks for having me. I would like to answer any question if you have.
Thank you so much, Ramin. That was a really fascinating talk, and it's especially useful to see such a clear going through of the various steps to implement a technique. So thank you so much for doing that. I have a question about optimal read depth for especially for very non-model organisms that can have truly dauntingly large ploidy counts. I know you mentioned that sometimes you have, or in general, you would have lower confidence in, as you were showing on the plot a couple of slides ago, where the coverage were very different from the inferred, for the different inferred haplotypes. And so I'm curious about whether in general, let's say you were working with a plant that was known to have very high ploidy in general, whether increasing the coverage over what you might, or the read depth over what you might expect would generally help with that? Or is that after a certain point of very high ploidy, is that sort of an insurmountable challenge?
From my experience, so both, let's not say both, actually WhatsApp is also like that. So WhatsApp, nPhase, HAT, which are the most recent tools for haplotype assembly.
Mm.
Like at every steps, they try to look for something with high support, and they try to take actions based on those supports. So like the more support, the more confident these tools are on taking the actions. And having a high coverage can actually help with that. So it will result in less errors. And that's a really good thing. So like they are more confident for taking actions. But you should always realize that like these tools are like the performance of these tools is so the computational time of these tools is quadratic to the number of reads, all of them. And the more coverage you have, these tools become slower, and at some point they will be unrunnable. Like you cannot even run them because they will become so slow that it's not usable anymore. So my personal opinion, based on the experience that I had with plants and also with yeast, 20x per haplotype is like the minimum. And I guess you already get good results with 20x, but 30x is a little bit better. It's like you are more confident with every action that you are taking. And more than that, doesn't change the results that much, but slow down the tools.
Yes, thank you. It's really helpful to have a rule of thumb like that to try to figure out where that, the worse than diminishing returns maybe start to come in. So yes, thank you. I just want to invite audience members again to raise any other questions, and to remind everyone also that the code that Ramin has shared is also really presented in great and really nicely annotated detail in the links that we have shared in the chat, and we'll also post with the video online. So let's just give it a minute to see if there are any more questions. That seems to be all of the questions, Ramin. So thank you again so much for such a clear and thought-provoking talk on the challenges and strategies for dealing with high ploidy. We really appreciate it, and best wishes with the next phases in your work.
Thank you a lot for having me.